Okay. So I do apologize if this is something that ends up confusing you. Just ignore it if you hate it. I got some complaints last time that they did not like something being called 2.5 structure. So if that is something that's confusing, just use the word protein domain or motif and go forward with that terminology. This was just for me to sort of orient you. We're going from somewhere between a beta sheet or an alpha helix to a full folded protein in tertiary structure. This is a common pattern of alpha helices and beta sheets, let's say. Okay, and so we'll look at that. So a polypeptide chain, or a part of a chain, generally in the range of about 50 to 200 residues. These lengths are not, you do not memorize these numbers, all right? Other than if you maybe solved a problem, got 300,000 residues, and then said, like, this isn't practical. Other than that, I won't ask you to memorize these. But so this is, a domain has a distinct organization of secondary elements. And we only talked about two, alpha helix and beta sheet. Okay? So proteins are formed from one or more domains, or often formed, okay, from one or more domains, each with its own distinct folding and diverse function. So the domains generally are a common pattern, a pattern that we've identified in a lot of proteins as we're analyzing them. And they have a particular combination of these secondary elements, and then they also generally have a specific function associated with them. Okay? So an example of these are the three domains of the Sarc protein. Okay? SRC is abbreviation for sarcoma. It is the type of cancer that is involved or associated with problems with the Sarc protein. Okay? So Sarc homology domains, or SH domains, there are three of them illustrated here. Okay? So we have the SH1 domain. You can see it's mostly alpha helices. There's several of them, 1, 2, 3, 4, there's several alpha helices. There's a couple beta sheets shown, and there's a binding site for ATP. Okay? SH1 domain is a tyrosine kinase. It can catalyze the transfer of one of the phosphates on ATP to phosphorylate some other residue. Okay? That's the function of that domain. SH2, as you can see, is a little bit shorter, a little bit smaller, lots of beta sheets, just this one alpha helix, and it has a site, again, for a phosphotyrosine. And then SH3 actually has a site to bind prolines. It's not super important to me that you memorize what these three domains can bind. It's more important to remember that they're distinct in their sort of pattern of secondary elements, and they thus have some sort of distinct binding site or some distinct function. Are they a kinase? Are they do they bind proline peptide sequences for some other mechanism? So you can get a lot of diversity in your different proteins by combining different domains. Okay. So the Sarc protein contains the three SH domains, the Sarc homology domains, as well as some unstructured linkers between them. So that's what, of course, all these little stretches of unstructured parts. And that allows, again, for flexibility. In order for ATP to come in here and bind, this binding site's going to need to be able to open and close. And we'll talk about ATP binding sites more chapter 12, but you can think of molecules that are able to bind ATP almost like breathing. They open and close really, really rapidly, 100 times a second, to be able to accept something in that binding site. So here's the overall Sarc protein. It has the three different domains hooked together by some linker, as shown here. So there's linker both within the domain and then between the domains holding them together. Each domain is stable on its own. So that's another key feature of a domain. It is stable. So rather than using just primary or secondary structure, it's often useful to describe proteins by their domain structure. Motifs are typically described a little bit more simply than domains. If you just really want to hate me, we could call motifs like 2.25 structure. I won't do that. Motifs are generally, you've probably heard in other classes, like helix turn helix or alpha beta alpha or something like that. So motifs a little shorter. Then we're talking about a little bit longer combo is the domain, and then a combo of domains comes together and we have our full tertiary structure. Okay, and so here's our Sarc protein here. If we're reading from N to C, we have the SH3, 2, 1 groups. But there are these other proteins that are exclusively made up, minus the linker pieces maybe being a little bit different of sequence. It's SH2, SH3, SH3. It's SH3, 2, 3. It's 2, 2, 1. So again, it's easier for you to describe that protein if you just think of it in terms of these bigger chunks, like chunks of regularly occurring sequences and folding. Once you understand this structure here, it's easier to just say, oh, hey, that protein has an SH1 domain in it. Okay. So a domain or a motif is a part of a protein chain that has its own distinct pattern of folding and its own distinct function. Domains will be stable on their own. It makes describing protein structures easier. And the Src protein contains the 3 SH domains, as well as the unstructured linkers between them. Again, you can just, this is a repeat slide. I called it 2.5 because this is where it fits in our overall list of structure.